And then in your top right-hand corner is your view. You can go ahead and push speaker view to hear myself and look at me, or gallery view. So it's up to you. And I'm going to launch a couple polling questions real quick. And we're going to start off with this one. What type of treatment are you currently taking? And I'm going to, since the recording does not pick up that, I will be reporting to you shortly. I'm waiting for the polling to come in, but if you guys have been to some ALK talks before, you probably understand what the answer might be. I'm going to end the poll and share the results. So alectinib is 69%, brigatinib is 17%, and lorlatinib is only 7%, which is weird. Normally it's 22%. So I'm stop sharing that.
Hey, we can't see that, Mark.
You got?
No. Did it say you were sharing it?
Yeah.
That's so weird, but that's okay. You can just tell us what they are.
All right.
I can see it.
I saw it.
You guys saw that? All right.
Sorry, just I can't. I'm sorry.
All right, Jenna, you're I froze you out. Here's the next question. What's your length of diagnosis since you've been diagnosed? All right, I'm gonna end the poll and share the results. So the majority, 46% is 1 to 2 years, 1 and under is 11%, 3 to 4 years is 23%, and 5 to 6 years is 11%, 7 to 8 years is 9%. So I'm gonna stop sharing that one. And my last question is, how many lines of treatment have you had? So how many TKIs have you taken? And you can also put down if you've taken chemotherapy, maybe some of you have taken immunotherapy.
No, it won't connect. I don't know why.
All right. I'm gonna end this poll, share the results. If this makes sense, also with the drug, 76% is 1 TKI, 2 TKIs are 18%, and 21% of us have taken chemotherapy, and 9% have taken immunotherapy. And that is my polling questions. Thank you so much. Please mute. And so I'm gonna send it off to Gina.
Big applaud for Mark. He's not feeling his best, and yet he still devotes his time to all of us ALKies. So thank you so much, Mark.
Well, I did have one more question. How many have had COVID before?
So
I got diagnosed on Friday. So
Mark's contagious, but thankfully not through Zoom. So we're really lucky to have him. As far as ALK Positive is concerned, I'm so excited to see all of you. We will be continuing to do our end of the year fundraising. So if you haven't participated in that, you can, if you go to alkpositive.org, just go underneath that donate button and you can either fundraise or donate through that. But we're really trying to raise as much money as we can so that we can really drive research for ALK positive patients. Today, I am so excited because we have an incredible panel here of ALK rock stars. We have Dr. Jessica Lynn, Dr. Justin Gainor, Dr. Richard Irwin, and Tony Addario. And so I'm going to open it up to Richard and Tony and just let you guys tell everybody about Alchemy and all of the incredible work that you do there.
Okay, Richard, you're sharing your slide. Hi, this is Tony Addario. We thought that it would be worthwhile to just give an overview of Alchemy, since some individuals may not be familiar with us. And then Richard's going to do all the heavy lifting with a couple slides we have. So Alchemy is a Addario Lung Cancer Medical Institute that we founded in 2008. It's a consortium of 30 academic centers and community hospitals in the United States and Western Europe. We view ourselves, as this slide depicts, working with various entities to help deliver studies that could be put together either by a researcher or a pharmaceutical entity or something that GoTo wants to do, or a patient group. And some of these studies we've developed and driven by ourselves. So we kind of see ourselves in the middle. We get funding from various entities. And Richard's going to talk a little bit about the studies that we have underway right now and some of the specific capabilities that we provide on these studies. Go ahead, Richard.
Thank you, Tony. Thanks, Gina, Mark, and everyone. So yes, Alchemy provides a number of capabilities to our investigator network. So often we look to our physician base, such as Doctors Gainor and Lynn, who are with us today, to come up with interesting ideas. We provide some of the legwork, getting trials designed, getting them up and running, overseeing them, helping to make sure that they are regulatory compliant by FDA guidances and typical standards of good clinical practice, et cetera. So we're a full service organization in many ways. As Tony mentioned, we have up to now 30 very prominent academic oncology medical centers, both the United States and ex-US, who make up the Alchemy network. So this is our brain trust, if you will. And these are the key opinion leaders that we collaborate with closely and help to advance their research endeavors. We've become, over the past 10 years, a real specialty group when it comes to running remote, completely digital, decentralized clinical trials. Many organizations, with the advent of COVID, had to pivot to this space. We did not. We've been doing this for a long time. We run these virtual trials very well and every day. So the advent of COVID really didn't challenge us operationally. And in fact, some companies came to us knowing that we were experts running virtual trials like this, which cut down a lot of barriers. Just a quick plug for the Alchemy team. Since you're all here, some of you in person, and I can see many of you, this is the Alchemy team. We are a group of dedicated, passionate professionals based in different parts of the country who are every day working on the trials that Alchemy is supporting. One of those, as all of you may know, is the spacewalk trial. This has been going on for a couple of years now. This is centered at Dana-Farber Cancer Center under Dr. Mark Awad and his team. Just to show you where we're at, we're about a third of the way through the planned enrollment. So we have over 100 enrolled in 300 that were originally put into the protocol as the target. You can see that in the middle of this chart, we had kind of a tepid set of monthly enrollment, and then we supplemented with social media campaigns and advertisements through Facebook and Google and YouTube and things like that. And we really saw a market increase as a result of those awareness campaigns. And we worked with the ALK Positive group to come up with a lot of those visuals and those advertisements. So I thank you all for all of those who sent photos in to us to be used in those campaigns. I won't steal the thunder of Drs. Lynn and Gainor to talk about the Enigma project today, but just to show you that it's a collaborative endeavor between Mass General, the ALK group, and Alchemy. Alchemy is specifically involved in upfront. My team will be screening and consenting potential volunteers for this trial. We'll be using a platform that's familiar to us to do that. It's hosted at one of our Alchemy network sites, and that's Vanderbilt University. Once we identify probable qualified subjects, and secure all the records required to do that. We'll pass that off to the Mass General team, who will be responsible for taking it from there, essentially establishing eligibility, enrolling the subject, managing the study thereafter. Alchemy will continue to be involved. In particular, we will be coordinating the logistics of collecting fresh sputum samples from enrolled subjects. We also host the hotline, the 888 hotline number, and the landing page where people can sign in and raise their hand to be a potential volunteer. Last but not least, as Tony said, we have ongoing right now and are always looking for qualified subjects who are eligible. This is the epidemiology of young lung cancer. It's a survey questionnaire. There is an optional blood draw that is part of that as well. We're looking for people who have been diagnosed originally with their lung cancer when they were under the age of 40. And this is a collaboration between physicians and epidemiologists at University of Southern California, Ohio State University, UCSF, and University of Hawaii. Alchemy is acutely involved and is running this on a day-to-day basis. And so with all of these Alchemy sponsored trials, you can go to our website, alchemy.net, click on the research tab, and you'll see an opportunity to learn about these trials. And if any of those seem interesting to you and you think you might be potentially eligible, we'd love to have you on board. I was quick. Thank you so much. I appreciate the opportunity.
Yeah, we certainly do. Thank you.
Yes, awesome stuff. And I wanted to say that when I first got diagnosed, I did not know what clinical trials were at all. And I thought that my very first study was epidemiology of young lung. And so I sent off urine and blood, and I thought all clinical trials were like that, where they came to you. And so that was one of the really cool things about Alchemy. I just wanted to kind of plug that and say that it really is incredible what you're doing.
Thank you.
We'll hand it over to Dr. Lynn, if you don't mind, just kind of give us an overview of Enigma and some of the incredible things that you guys are doing.
Yes, hi everyone. Good to see you all again through this session. We know it's been a really hard year, and especially these few weeks with cases rising again. So you're all focusing on taking care of yourselves and wishing all of you a safe holiday season. So for today, we're focusing on the Enigma Plus protocol, as you heard, which is abbreviation for elucidating novel immune and genomic markers for ALK positive. And I think many of you on this call are probably familiar with the kind of background for the treatment of ALK positive lung cancer. I'm going to spend just a couple of minutes summarizing the most important concepts that are relevant for the rationale of why we launched this protocol. So as most of you know, we now have 3 generations of ALK tyrosine kinase inhibitors that have received FDA approvals. I've shown here many of the approved ones, and Encertinib is the only one that is not technically approved in the USA. That's a 2nd generation ALK inhibitor. And all of these are highly effective ALK inhibitors. These days, when you're newly diagnosed with advanced ALK positive lung cancer, most of the patients will end up starting on a 2nd generation ALK inhibitors, most commonly alectinib. And then you may receive another ALK inhibitor, most likely at least involving a 3rd generation ALK inhibitor, lorlatinib. And then at some point in the disease course, there may be treatment with chemotherapy or clinical trials. Lorlatinib did receive FDA approval for 1st-line indication as well recently. However, I think it's important to remember that we still have patients, as some of you will be familiar with, who were diagnosed several years ago. And sorry, I am getting some feedback. I don't know if some of you have unmuted. If you can please mute your speakers. So we still have patients that were diagnosed several years ago when crizotinib used to be the standard initial therapy. And these patients will have received crizotinib as the initial ALK inhibitor, and then may have received 3, 4, 5 different ALK inhibitors since then. So that is relevant for how we think about what we're seeing in terms of the resistance landscape and how to overcome them in clinic. The thing is that the rapid pace of the evolution of ALK inhibitors in the past decade has been really good to see, but it also brings with it some challenges in terms of keeping up to date with understanding what causes disease progression on these drugs and how to address them. So work by our group and several others have told us that resistance to these ALK inhibitors arises by two main branches of resistance mechanisms. So the first branch is the on-target resistance mechanism, and largely that is mediated by acquired mutations in the ALK kinase domain that impact drug binding or induce some sort of conformational change where the drug is no longer able to work on the target as effectively. The second big branch is the off-target resistance mechanism, and that can be mediated by various mechanisms, most commonly involving activation of another signaling pathway that allows cancer cells to continue to survive and proliferate even when ALK is effectively bound by the TKI and inhibited. Sometimes we also see changes in the phenotype of the cancer cell, where what used to be adenocarcinoma or non-small cell lung cancer, more broadly speaking, is transformed into now small cell lung cancer, which has fundamentally different biology and ways that we think about how to treat it. This is a good example of how resistant biopsies have really helped us understand the biology of these resistant tumors. So this is work by Dr. Gainor, who's on this call today, that's been updated from our Mass General resistant biopsy series, where we're looking at tumors that are taken from patients progressing on crizotinib, ceritinib, alectinib, or brigatinib. And it is through gene sequencing and gene analysis of these resistant tumors that we were really able to understand how each of these ALK inhibitors is associated with a really unique distribution of ALK kinase domain mutations that cause resistance to these agents. We know that there are certain mutations that are common across the board, like the one highlighted by red color here, which is an ALK G1202R resistance mutation. But then we know ones that are specific to certain ALK inhibitors. For example, for alectinib, which is the one that most of you are on right now based on the poll that was performed at the beginning of this session, the most common mutation is G1202R, but another common mutation involves this I1171 residue. And with this mutation could still be sensitive to the other second generation ALK inhibitors such as ceritinib and brigatinib. These efforts were really important in helping us think about when we see these resistance mutations on biopsies in patients, how do we then structure our thoughts about what might be the viable next line treatment options? We also have some understanding of what causes progression on lorlatinib as well. And so again, this is a series from Mass General where we've looked at 45 lorlatinib resistant tissue biopsies. And you can see that in about a third to a quarter to a third of cases, there are these ALK mutations that are found in the resistant biopsies. And what you're looking at with the slashes in between the different mutations is indicating that there are these two different ALK mutations that are detected concurrently on biopsies. And then there are cases where we see 3 different ALK mutations concurrently. And often when we are able to do more in-depth studies, we're able to confirm that these mutations are occurring together. And that's what's causing the tumor to escape lorlatinib. We've also been able to do additional studies like whole exome sequencing and more complex computational analysis that have told us that these mutations are often occurring in sequence. So for example, there may be one ALK mutation that's acquired when you are on a prior ALK inhibitor like crizotinib, let's say, or alectinib, but then you go on lorlatinib and you may yet acquire another mutation on top of that. And that's what causes the cancer to progress on lorlatinib. And while this has been a great advance in thinking about, well, what are the options that we need to think about, what are the novel therapies we need to develop for lorlatinib resistant disease, you can see that the numbers here are still limited. And it makes us wonder, well, what if we could look at a much larger number of tumors? What if we could get tumors from a larger patient panel? Could we learn something additional about not only the spectrum of ALK mutations that can confer resistance to lorlatinib, but also non-ALK resistance mechanisms as well? Another background fact that's worth pointing out is that we know very well at this point that these immune checkpoint inhibitors like pembrolizumab or Keytruda or nivolumab or Opdivo are not really effective by themselves in ALK positive lung cancer. And so some of you know that in general, when we think about general non-small cell lung cancer, immune checkpoint inhibitors have really become one of the standard therapies, really important therapies that we reach for, even at initial diagnosis. However, there have been multiple studies at this point that have demonstrated that the response to these drugs is very limited and minimal in patients with ALK positive lung cancer. These are data from a study that Dr. Gainor performed actually a few years ago, looking at clinical benefit from checkpoint inhibitors in patients with ALK rearranged tumors or EGFR mutant tumors, which we think of as kind of similar in their biology, as compared to tumors without ALK rearrangements or EGFR mutations. So here you can see that while the response rate to checkpoint inhibitors was around 20%, in those tumors without EGFR, or in patients with tumors without EGFR mutations or ALK rearrangements, in patients with EGFR mutant lung cancer or ALK positive lung cancer, the response was pretty much close to 0. And then the progression-free survival was also significantly shorter in patients with EGFR mutant or ALK positive lung cancer. So that provides a second rationale for the Enigma Plus protocol is that when we look at these larger number of patients, not only can we look at the genome landscape and understand better the evolution of resistance to the ALK inhibitors, but also start to study more in depth the immune landscape of the ALK positive lung cancer and what are the different components that play into minimal benefit from the available checkpoint inhibitors, and what are the components that could potentially lead us towards novel immune-based strategies that could become an option for our patients with ALK positive lung cancer. So that led us to think of this web-based patient-driven platform in collaboration with Alchemy, as you heard about earlier. And the overall goal is that with this platform, patients who are not necessarily seen at Mass General, who are across the nation in different states, who are not able to travel to Boston, are still able to participate in our streamlined research process, where you go online and provide informed consent after learning about the study, and that initiates the process of our being able to collect clinical data as well as tumor samples with which we can then perform genomic analysis and immune profiling. So this is the outline of objectives, main objectives of the study. As you heard, we're aiming to look at clinical data from at least 100 patients with advanced ALK positive lung cancer, and ideally tumor specimens from a majority of the patients in order to characterize the immune phenotype and genomic landscape in ALK positive lung cancer. There will be secondary objectives of looking at clinical outcomes of different treatments, including ALK inhibitors and immune checkpoint inhibitors, either together with chemotherapy or by themselves. We'll be looking at different ALK resistance mutations in the resistant tumor biopsies. We'll be looking at non-ALK gene alterations, and also looking at different factors that play into tumor immunophenotype that I'll go into in a little bit. As you heard about, there was a lot of patient interest from the get-go when we first envisioned this protocol, and you can see here some familiar faces of patients that kindly provided their photos to be included for this social media outreach. And on these flyers, you can also see the portal address where you can log on to in order to learn more about the study and express your interest. So this is the page that you see when you first enter this website. You can see here a little paragraph about the study. If you click on the plus sign, you'll read about study purpose and what you can expect in the study once you express interest. And at the bottom, there is a contact form, which looks like this on the right-hand side, where you provide very basic information that allows us to have what we need in order to get the informed consent going. So name, address, email address, phone number, best time, date, time for contact, and so forth. Here, what I wanted to highlight is that to be eligible for this study, The criteria are very loose and open, and intentionally so, because we want to encourage the participation of patients as much as possible. Basically, you need to be an adult with advanced ALK positive lung cancer diagnosis and be willing to provide clinical and medical information to the study team. And you have to be based in the United States. So you have an idea, a general idea of what will be done once you provide consent to the trial. Here are the samples that will be collected once you provide consent. So again, no knowledge of the tumor availability is not required for you to consent to this protocol, okay? So you, as long as you're interested in providing your clinical information and tumor sample if available, you can consent to this study, and then our study team would look into whether there is sufficient tissue available for us to collect. The requirement for tissue is minimum of 5 unstained slides or a maximum of 20 unstained slides. And what we do is, once we've secured a minimum of 5 unstained slides and we know we have at least that amount of tissue in our hands, then that launches the Alchemy teams contacting you with the saliva collection kit. So that's mailed to your house, and there will be instructions on how to collect the saliva and mail it back to our study team. Here is a very general idea of what kinds of studies can be performed with the tissue that is collected. So an easy starting point, right, is if there's enough tissue to do comprehensive gene sequencing analysis. And that can give us a good starting point for a lot of information about the gene landscape of the tumor, both treatment naive and resistant tumors, looking at mutations in the ALK gene, as well as other cancer-related genes. That can also give us a sense of the overall tumor mutational burden, which can be relevant for thinking about immunogenicity of the tumors. And then we can also do other studies to look at how different signaling pathways may be activated or not activated in the treated tissue. And then I'll give you some other more specific examples in these next few slides. In terms of looking at the immune landscape of the ALK positive lung cancers, we already know that there can be several factors associated with response to PD-1 inhibitors. And this slide is courtesy of Dr. Gainor. You can see that they can be broadly categorized into host factors, factors that are intrinsic to the tumor, to the cancer itself, the tumor microenvironment, the environment surrounding the tumor, with infiltrating immune cells, and then clinical factors with regard to exposure to prior carcinogens and steroid use. Some of these can definitely be looked at with the tissue that's collected, including, as I mentioned, tumor mutational burden. We can work on whether we can predict neoantigen landscape. We can definitely look at PD-L1 expression pretty easily with minimal amount of tissue if it's not been done already, and looking at other infiltrating immune cell composition as well. Here is an example of the kind of data that we can put together at an aggregate level once we have a good enough number of patients that are participating. This is from a separate project, and it's looking at not specifically ALK positive lung cancer, but really general non-small cell lung cancer. And you can see that here at the top, we can look at the mutation burden in each individual tumor. And then for each line, you are looking at different factors for each patient. So you can look at the response to immune checkpoint inhibitor for that patient, their smoking status, what was the histology of the tumor, what was the PD-L1 expression, and what was the landscape of other co-occurring mutations in the tumor that may be relevant to how we think about response to PD-1 inhibitors. And all of these factors can be looked at in aggregate for each patient and then across all of the patients, and that can give us a lot of information that can be very valuable. In terms of when I mentioned looking at other immune cell types that may be relevant for thinking about benefit from immunotherapies, we have novel technologies that are being developed to allow concurrent assessment of these different immune components using minimal amounts of tissue. So one example is using a technology called immunofluorescence, where you can stain for different types of cells using, for example, one slide of tissue, which is a huge advance over having to require multiple slides to look at each slide for each different protein. And so if you look at this picture here in the middle, you can see that different colors can be used to label different relevant proteins in the immune field. And looking at overlap of different colors can give us a sense of whether there are cells that are, whether there's a co-expression of different proteins as well. Ultimately, once we start gathering some data, we're thinking about presenting in a portal like the CBio portal, where not at an individual tumor level, but at an aggregate level, what we're looking at in terms of what kinds of histologies we're seeing, what kinds of mutations we're seeing in aggregate, what percent of tumors seem to have immune cell infiltration versus not, and so forth. So we hope to be able to share the data at some level with the ALK positive and broader patient and researcher community. So that concludes my overview. And
wonderful. How exciting about this study. And thank you guys so much for putting this on. There are a few questions. One big question, and you know that this is an international group. So we were just wondering, is there ever going to be an opportunity, or what are the limitations as far as, you know, bringing it internationally or making it a worldwide study?
Yeah, that we are aware that there is some interest in knowing whether this can be broadened even further to international participation. We would love to do that. Unfortunately, there are some regulatory hurdles. As some of you know, even just for this study for a national patient population, there were a lot of hurdles that we did have to jump through that led to some delay in the activation of the protocol. We would love to continue to think about how we could enable international study. I think that would take a lot of time and funding, and yeah, we are hoping to at least get this going, implement at the national level, and get some patient enrollment and see what the preliminary data shows first.
Great. Is there any limitation as far as, so I think it will be easy to get 100 ALK positive patients. We're all excited and we really want to help advance research as much as possible. Is there any limitation or any reason that the limited amount of tissue that might be at our institution shouldn't be given to this study? Is there a reason for it to stay at our institution, I guess is the question.
Yeah, that's a good question. And I think that really is a hard question to answer at a kind of a blanket level. You know, there will be some clinical trials, for example, that do require tissue. But in considering that, we did put a limit on the number of slides that would be collected from each case. Yeah.
And I would add there, as Dr. Lin showed, you know, tumors can evolve over time. And so our view is, you know, we think of getting, you know, repeat liquid biopsies as well as, you know, repeat tissue biopsies as a standard of care in patients who are progressing on targeted therapy. And so I think because of that, because we do know that that can then inform subsequent decision-making, you know, the tumor isn't necessarily static. And so if someone was then thinking about participating in another clinical trial, I would say most of the time we'd be wanting to perform another biopsy if safe, if feasible.
That makes sense. Dr. Gainor, you've been in this space for so long. What are your hopes to get out of this study? What is the big hope that you? Yeah,
first, I want to say a number of thank yous. First to the ALK positive community for really supporting this. As Dr. Lin mentioned, you know, it has taken a while to get off the ground, but we're really excited about it, and we're so glad for your partnership. I also want to thank Tony and Richard really for you know, helping us all along the way. This is I know they share our excitement, and I think they were certainly ahead of their time in terms of creating this you know, virtual platform that I think will really enable a study like this. And then I also want to say a huge thank you to Dr. Lin, because I think no one else can appreciate how much work went into this. It may seem like pretty straightforward, but the amount of operational, logistical, regulatory work that went into it, as well as the science piece of actually, how do we prioritize certain analyses, and how do we even extend the reach? Because we think part of this is an infrastructure, right? It's an infrastructure to then facilitate downstream analyses. And so I think some of the exciting pieces, we mentioned, she highlighted a lot of the genetic work, and you can think about genetics on, relatively narrow focus, or we can do really complicated genetics where we're looking at every single gene in the human genome. And, she's been able to now partner with the Broad Institute and IBM to actually allow us to then do whole exome sequencing, which is really extensive sequencing of specimens that come in through this platform that will give us the most robust assessment, and actually looking at it with world-class geneticists, Gadi Getz, who is really, if those of you who have heard the TCGA, the Tumor Genome Atlas, he was one of the original architects of that. And so to have his team looking at this, as well as getting funding through that to support some of the genotyping, I think is huge. So I think, again, huge thanks to Dr. Lin, because this has been a monumental lift. And I think we're excited about it because, like I said, one, I think it is. We get the point about, the US is only a small portion of the global community that's affected by this disease. I think first, there's also, we need to assess the feasibility, right? This is a completely new way of looking at ALK positive lung cancer, trying to leverage the community. We recognize that not everyone is able to travel to Mass General, right? And so I think this it does also come down to, I think it helps promote equity too, in terms of basically, everyone, potentially being able to participate in this. And I think I'm excited that our technologies are also constantly evolving, and that we have partnered with other world-class scientists, not just Dr. Getz, but world-class immunologists, Nir Hacohen, who heads the tumor immunology program at Harvard and MGH, as well as our expert pathologists. And we're really thinking about, these are the next tier omics that I think we need to employ, because, genetics will only get us so far. And now we need to look at these next generation tools to try to uncover, particularly that other half of tumors that don't develop resist ALK mutations. They have other mechanisms of resistance. And there's a lot we still don't know about that. And I think this study will be pivotal in helping us sort that out.
Great. There's quite a few questions coming in. One is from our friend Ravi, and he wants to know kind of if it shows up that you have ALK, but you don't have any sub mutations, is this something that you can participate in?
Yes, absolutely. Because like just mentioned, I think that's an area that we absolutely need to learn more about. One of our colleagues, Ibiaja Gogo Jack, had recently described, she's also at Mass General, met amplification as a mechanism of resistance, but that we know is a relatively small fraction. And we need to figure out what those other drivers of resistance are, because it's not just describing them, it's then us thinking about tools to then therapeutically target those vulnerabilities. So absolutely, a patient like that would still be eligible here.
So, and that kind of brings up a question of, does this tissue even have to be brand new tissue? Can this be old tissue that's been in your hospital setting for a while.
Absolutely. And that's actually really valuable to have a series of tissue from different time points if that's available, because it also allows us to look at not only a single time point capture of the biology of the tumor, but look at how it really evolved over time as the patient was receiving different therapies. So that alone can also add valuable information. So archival tissue is what we're looking to collect from different time points since the diagnosis.
Great. So if you are only currently having brain metastasis, it would still be beneficial to send our tissue for maybe from the lung from when we were first diagnosed or something like that. Is that correct?
Absolutely. Again, we're interested in a broad array of different cancers. So even cancers that have just brain metastatic disease may have something valuable to offer in terms of resistance biology that's unique to brain metastasis. So as long as there's tissue available, we'll be working to collect it.
Awesome. In our group, we have people who have ALK positive cancer that is other tumor types, but I think I saw on your slide that it's only for non-small cell lung cancer, Enigma. Is that correct?
That's right. We're starting with lung cancer for now to focus on that because that's kind of where we want to focus our energy on understanding the resistance mutations landscape and the immune landscape first. Now we are learning that a lot of lessons that are learned from lung cancer could also be extended to other disease types. And depending on how this study evolves, there could be amendments in the future where we enroll other disease types, but we felt that lung cancer was a good kind of disease type to start out with.
We had the question, and maybe you can answer this. Is there any research being done on TP53?
So that would, Justin, go ahead.
Oh no, the short answer is yes. And maybe it's worth taking a minute to step back about the role of TP53, or sometimes we shorten and just say P53. And there are two major types of genetic alterations that we think about when it comes to cancer genetics. For much of our discussion tonight, we've been focusing on ALK, which is an oncogene. So this is something where all it takes is that's a gain of function. So that is a gain of function alteration. So where basically in the setting of that rearrangement, it's sending constant growth signal. So gain of function. And it only takes one. So one hit, one of the two genes. But by contrast, P53 is a tumor suppressor. So that's something that is, it's known as the guardian of the genome. There, it's a loss of function. So it's so mutations that basically causes it to lose its activity. So there you need two alterations for it to fully lose effect. The challenge is that things like P53 are commonly like passengers. That is, that they don't drive the original formation of the cancer. They come along for the ride and they make the cancer grow faster because it helps protect the genome. And so when that's altered and you lose that function, the cancer can then acquire more and more mutations. It's basically losing some of its proofreading mechanisms. And it's much harder to replace something than it is to block something that's aberrantly activated. So that's why in oncology, when we talk about most targeted therapies, we're talking about inhibitors. So we're blocking overactive signals. We have a much harder time at replacing things. So I think for us, tons of research going on P53, and that will be something we'll look at. The role of these other mutations and how do they modulate responses to ALK inhibitors.
Thank you so much. One of our friends, Rick, he can't quite get on the chat, but he was wondering, is there a certain time limit that our institutions hold our tissue? So his last tissue that he had was like 7 years ago. So is there a certain time limit?
Yes, there can be, and it can vary across institutions.
Uh-oh.
Okay. Did you hear that some institutions, it's institution dependent, and some institutions, for example, will hold for 15 years max and then discard. So that's really variable institution to institution. 7 years usually should be fine, and rest assured that our study team would look into all of this, right? None of this burden is on patients. It's the study team that would contact the institution with the consent and the medical record release form to check into what tissue is available and how much we can collect.
One of the things that was really exciting for us was spacewalk, and that was one of the things that we were really excited to be participating in. And we knew that when we participated in spacewalk, we would give a liquid biopsy and we would get the results of that. So we were wondering, will the patient actually get the results of this archived tissue next-gen sequencing?
Yeah, so many of the studies that we're looking to perform are assays on a research basis that's not technically CLIA approved at a clinical level. Some of the assays, for example, that are really novel, looking at multiplex immunofluorescence, are just nascent in the research laboratories. So we wouldn't be able to give back clinically implementable information to the patient, but what we really do want to do is we really appreciate all of your interest and the generosity in participating in the study, and we definitely feel it's very important to share the knowledge that's gained with everyone in the community. And so what we're really hoping to do is to be able to present the knowledge that's gained in terms of the mutations and the mutation burden and the immune landscape in a patient-friendly format at the aggregate level, and present that to the public domain. And of course, in meetings like this as well.
And part of that is we don't know for most of these tests that we're performing, we don't know the meaningfulness of it right now on an individual patient level. It's really in aggregate where we start deciphering, what are the cutoffs? How do we score it? This is truly in that discovery phase, as opposed to the standard next generation sequencing platform. So I would view it, just seeing some of the questions, what is the difference between Spacewalk and Enigma? I would view these as complementary, right? We heard early on about kind of the enrollment in Spacewalk. So absolutely, we would say we'd encourage people to participate in both. We work very closely with the folks at the Farber and Dr. Watt and colleagues and collaborate a lot. And so I think, again, I think this is something where we can leverage both sets of data. I think the other piece is, we also view this as, part of it is, getting data out there in the public, one, to recognize, the amazing contributions of our patients, but secondly, also to have researchers around the world also do their own downstream analyses and also look at data in a different way or answer different questions, things like that. And then the last piece is, even when we're doing genetics, we're doing the genetics, as I mentioned, we're doing this whole exome sequencing. It's not in a clinical lab though. That's for several reasons. One, we can get much greater breadth. We can look at every single gene, but it also we can also do that in a much more cost-effective way. So non-clinical genotyping, we can do for 1/5 of the cost as well, and so that also it just extends the reach, and we get more data from it. So I think those are all factors as well.
Right. In our ALK positive group, we have quite a few ROS1 people. That's our favorite cousin. And so we were just wondering if this study would be open to ROS1 as well.
That's another great question. You know, this particular study is focused on ALK positive lung cancer. However, I think you bring up a great point, which is that let's say the study goes very smoothly and we learn a lot of valuable information, which is what we're hoping for. I think the concept and the idea could certainly be extended to other important molecular subsets of lung cancer, including ROS1 positive lung cancer. And that would be the best case scenario, right? That we not only learn lessons specific to ALK positive lung cancer, but also that we learn lessons about implementing this sort of a study that's a web-based consent study. I think over the next few years, we're going to see more and more of these studies, hopefully, because I think they're becoming much more important in broadening the reach of the clinical research studies and galvanizing on that.
Maybe we can get Tony or Richard to weigh in there too. Just what have you seen in terms of scalability? You're now, Alchemy is now coordinating a number of different studies. Can you talk about scalability and kind of what you've seen in terms of kind of bandwidth within Alchemy?
Yeah, I'll let Richard answer that as well. But we are a virtual organization. Our studies are virtual. We bring consultants on to help with various studies for the length of the study. So as we need more resource, we can bring them on board. So, you know, I'm sure there's an infinite number that we would have to grow, but the fact that we're virtual, we can do this remotely. We have the experience, as Richard said, we've been doing this from our first study, which was a tissue collection study, Castle, where we were the first entity to do specimen collection of stage 4 non-small cell patients, 14 years ago. And so we have the bandwidth. We would need the appropriate funding to get to many more additional studies, but we're eager. We're always looking for more things. We have another study that hopefully gets launched in Q1, and that's for KRAS, which is very exciting for us since there's been so little things going on in KRAS. Richard, you're the one that has to run it. I can make all kinds of promises.
Well, I think you said it well, Tony. Nothing really to add to what you've said. I just want to also point out, in a nod to Doctors Lin and Gainor, that particular protocol is written smartly in that it is designed well to be expanded into different cohorts and possibly different regions quite readily. So that was always part of the forethought. So I give them credit for, writing a protocol that wouldn't be difficult to include different tumor types and different organs as well.
We're ready, Justin.
Yay, yes, keep it going, because I think the more that we know, the better. Knowledge is power. One of the questions on here is, can you get tissue from Foundation One instead of from your institute? Can you guys use our tissue from Foundation One?
Yes, so the tissue, how it works behind the scenes is the tissue is sent from the institution where the biopsy is performed to Foundation Medicine for sequencing. But whenever other study teams or other institutions request tissue, it's requested directly from the original institution. So the request would be made to the original institution. Ideally, there would still be some tissue left over where the tissue originated. Sometimes if the sequencing had been performed pretty recently, then sometimes there has to be a request made back to Foundation Medicine to send whatever is remaining back to the institution, but that would all be kind of worked out behind the scenes as well.
I see one question in the chat that I want to make sure that I address, which is, someone's asking, if I have biopsy tissue, but it has been sent for clinical genotyping, should I do that? Or so I would say Dr. Lynn and myself, we always put clinical care first. So, so absolutely, if someone's had a resistance biopsy, we would want that person having their routine clinical genotyping. We don't view this as a substitute for that. So, and that's how I would prioritize things. But what we're talking about is kind of what's left after that. The remaining archival tissue, I think, would be well positioned, for use in this study. I did want to make that explicitly clear.
Thanks so much. Somebody asked, how do you get the tissue out of your institution? And I think that you explained it pretty clearly. I think I was going to just go ahead and put it up there, but I think we just begin by filling out the I guess contact sheet. Is that correct?
Yes.
It's
So
the-
just Richard, go ahead. You can take this.
Yeah. If you go to the alchemy.net page for Enigma, and someone put it in the chat earlier. I think Gina, you put it in the chat earlier. There's a as you know, there's a form that you can fill out, simple form just with contact information to raise your hand and indicate that you want to talk about this trial and it's interesting to you. The Alchemy study team will contact you within 48 hours, and I want to thank the 3 people already who have sent those in the 30 minutes that we've been on. So you know who you are. Thank you so much. You'll be hearing from one of my team members within the next 48 hours, and that's the way to get the process started.
Keep them coming, because we know that the more that we can contribute this data and the more that we learn about this disease, the better off for all of us. So thank you guys so much for doing this. We're coming up on the hour. I just wanted to open it up for any final remarks that you guys might have.
I'd say thank you for the opportunity to present and to talk about the studies that we have underway. We definitely appreciate it. And we appreciate your help in all the social media that we've put on for the various ALK studies.
We are so thankful for you guys.
Yeah, I just want to echo that. Thank you so much. As Dr. Gainor said, we're so appreciative of the Alchemy for the collaboration and all of you for your generosity and always thinking about how you can contribute yourself and everything to these research studies to advance the knowledge in general. We know that takes a lot of generosity. So thank you so much. And I just want to say again that I really hope that all of you and all of your loved ones stay safe and have very safe and peaceful holidays.
Oh, thank you so much, Dr. Lynn. Thank you so much, Richard. If you guys want to take yourselves off of mute, feel free to thank our guests.
Thank you. Thank you very much.
Thank you.
Thank you.
Thank you very much.
Thank you. Happy holidays. Merry Christmas.
Thank you.
Happy holidays.
Thank you.
Thank you so much.